Well, I guess whilst I wait to see if Aaron can find a place that has better internet connectivity, I guess I will welcome everybody to the to the first and hopefully first of many Bioconductor developers forum, teleconference, call it what you will. So I thought I would just give like an introduction as to what I was hoping these meetings would be, and then a little breakdown of what we're going to do today, and then we'll see what happens. But it's all a little bit experimental. So the concept is hopefully that this provides another mechanism along with things like the developer mailing list and Slack and things for people who are developing our code, whether they are in the BioC core team or they're just getting started with their first package, to discuss concepts in software development, either general concepts or specific things about individual packages or some of the core infrastructure, how best to use it, new developments, that kind of thing. The way I sort of envisage it taking is about an hour once every month. Preferably there'll be sort of maybe 2 to 3 topics we discuss during it. My idea is that one person will basically present a short presentation, 10-15 minutes, followed by a similar amount of discussion about that topic. That's the idea. So I came back from holiday 2 days ago, so I didn't chase up lots of people to present today. So the first presentation will be by me. Hopefully, if he's managed to get internet connection, the second presentation will be by Aaron. And then I have a couple of thoughts for other things we should discuss today. So there's an agenda in the Slack channel if people are interested in looking at that. Mostly about how we select future topics and what time we should do this meeting, because we're excluding everybody in Australia, I guess, at the moment, by doing this at 4 o'clock in the morning. So whether we should rotate times and that kind of thing, I don't know what the best strategy for that is at the moment. So I'm hoping everybody can hear me. Can a few people reply and just acknowledge that I am saying something here?
Yes, I can hear you.
Thank you. Perfect. A few thumbs up, that's good. That's what I like to see. In which case, I'm going to give the first presentation in the sort of style that I kind of envisaged, which is that it's going to be about 10 minutes long. There's a little bit of background concept as to a bit of context as to why I'm talking about it, and then hopefully some leading questions at the end that we can discuss. And it's going to be about the biomaRt package, which is one of the packages that I maintain. So hopefully you enjoy this, but if you don't, it's going to be the last time you hear this style, and we'll adapt things going forwards. So fingers crossed I can share the screen properly. If not, there's a link in Slack to the presentation. So is that showing up for people? Get some more feedback.
Working great.
Awesome, excellent, great, thank you. So, yeah, I want to talk about biomaRt, the package, which is in Bioconductor. And before you can really talk about biomaRt, the package, you have to talk about the difference between biomaRt and biomaRt, which is always I'm sure this was an excellent design choice when coming up with the name, but it's really frustrating when giving a presentation to try and distinguish the two. So there's capital B, capital M biomaRt, and then a capital R. And we'll actually start with biomaRt with the big B, big M. And I think actually most users tend to conflate the term biomaRt with ensemble, and they think of the two almost interchangeably. And a lot of the time when people ask me questions about it, really they're referring to ensemble data rather than biomaRt as a concept. But actually, biomaRt doesn't have to equal ensemble, right? If you dig around on the internet, there are actually other examples of data being served up with the biomaRt suite of software. Most of these are now pretty small, but there's Wormbase. My favorite is the International Potato Center. But there are other examples of biomaRt, because that software has actually been around for nearly 20 years now. So it doesn't necessarily equal ensemble, but in reality, the vast majority of the time, when people are talking about biomaRt, they're talking about getting data from ensemble. Those other databases, and 10 years ago there was probably 25, but most of them are now either dying or they've been absorbed into Ensembl. So others specific mutation databases and things are now basically incorporated into Ensembl, or they're moving to other platforms, which you might understand why they're doing that as I go through this talk. So in reality, we do generally mean getting data from Ensembl with the biomaRt suite. And then biomaRt with a capital R is basically an R package for querying these biomaRt instances programmatically. And originally this was developed by a guy at the EBI in 2005, and he basically maintained the package for about 10 years, and then I took over about 4 years ago. And this historical requirement to look after many different databases means there's kind of a split in the package between things which are Ensembl specific and things which are generic for accessing other databases. But now most of the defaults are also Ensembl, and there's this kind of weird double syntax where you can you do every operation in two different ways depending upon whether you use the Ensembl specific code or the generic code. And I'll be interested to know whether people think it's worth refactoring that or not. And just to kind of demonstrate, whilst this isn't a particularly exciting package, it's been around for a really long time, kind of what a cornerstone it makes. There's actually 118 Bioconductor packages that list biomaRt somewhere in the description. So there's an awful lot of packages that depend in some way on biomaRt functionality. And that doesn't go into the ones on CRAN, which also access it. And you might think that because all those other databases are dying, maybe they're historical packages that use it, but people don't actually need it anymore. But actually, if you look at the number of people downloading it, it goes up every year. The number of questions I get about it goes up every year. And it's not just kind of automated downloads. People are citing it still. You get several hundred citations every year for the package. So I think it's still really being used. And I kind of just wanted to use these slides to emphasize that whilst it's not a hot topic, there is a lot of other software and people that depend on biomaRt. So looking after it is potentially, if we don't do that, it's potentially damaging to the kind of overall ecosystem in Bioconductor. But recently there's been quite a few issues. So basically since Ensembl 96 came out, which was April of this year, basically queries just started failing much more frequently with this kind of error 500, which basically means server error. It doesn't give you any information about what happened, why it's gone wrong, just there was basically a massive spike in people asking questions either to me directly or on the Bioconductor support forum with this error in the last kind of 6 months. And anecdotally, it seems that like larger queries fail more frequently, which is frustrating to a user who has something that runs for a long time and then fails. They basically have to go right back to the start and run it again with no guarantee it will work the second time either. And typically the mirror sites for Ensembl were a bit more responsive, so my standard answer was to basically tell people to use the mirror sites in America or in Asia, but that more felt like it was fixing a symptom rather than a robust response to this. And also in a kind of unrelated thing with the most recent release, there was an unexpected renaming of a crucial attribute. So in this case, when you want to access EntreeGene, you have to use EntreeGene_ID instead of EntreeGene, which had been there for years and years. And basically both of these changes are kind of breaking errors or changes to both biomaRt as a package, but also to the downstream packages that use it. Anybody that used EntreeGene as an example in their vignette, they immediately broke. And similarly, if you can't predict this error 500 thing happening, it's quite hard to have a stable package. So I've made a few package changes in the last month or so to try and identify, to try and mitigate some of these things. And I'd be really curious to know whether people think these are sensible approaches to have done. So the first thing that I do is that the queries people submit are now broken down into batches. And the result of each batch, so that each batch is submitted separately and the results of each one are basically written to the temporary directory. So if you rerun the query, instead of arbitrarily getting somewhere and failing again, it will automatically pick up where it left off. So each time you rerun it as a user, whilst it's frustrating, it fails, you're more likely to get to the end, basically. And this isn't persistent. It will, as soon as you quit the R session, all of those temporary files are deleted. Then if a query actually completes, I now use BioC file cache to store the complete result table that comes back. And I identify that basically using a hash based upon the parameters that are passed to the calling the query function. So that means that those are persistent. If you rerun it again, you will just load the results straight from disk. Ideally, I think you would obviously hope that a user would run it once and then save it somewhere or save the results and things. In my experience, people don't tend to do that for things that run for a couple of minutes, which is what a biomaRt query typically runs for. So they're quite happy to run it again, but not if it fails multiple times. And I've also re-enabled the mirror redirection for based on your geographical location. So this is something I turned off in the past because I found it annoying that people would not go to the host that I would go to based in Europe. Debugging things was quite difficult that way. But actually, if it seems like the mirrors are more robust, and maybe this is actually a good strategy to implement to allow people to be geo-redirected. However, this doesn't actually solve the build failures, right? It's slightly more useful for a user. They can press rerun and it will pick up where it left off, kind of thing. But I have no idea whether if you run it once in the build system, will the cache that gets, if it completes, that's great. There's a chance it won't, but if it does, will that cache remain between builds? Is it a valid test if the cache exists already? I'm not sure, and those are kind of open questions. I'd love to know, like how the build system deals with those kinds of things. Do files remain on disk between runs of the build? And I've also been in conversations with Ensemble themselves to try and understand this, right? So they basically don't have a concrete answer, at least not one they're giving to me as to why it's happening. They basically said that the problems are affecting the web interface too, which is obvious if you try and use it. That you time out in a similar way, kind of thing. You just get a spinning circle. And they basically said it was due to their infrastructure struggling to cope with the number of genomes that are now in Ensemble, without really having a concrete answer as to what they're planning to do about it, at least in the short term. One thing they did say is that basically biomaRt with the big B and big M is effectively dead, right? They keep running it, but nobody's going to maintain the code. Nobody's going to look after it. So if this is an issue with that software suite, nobody's going to come up with a more effective way of modifications to that itself. And one specific thing they did mention is that massive, and I don't know exactly how big massive is, but massive R queries, they've noticed that these sometimes cause issues. I know there was one person on BioStars who was trying to submit 17 million SNPs in batches of 500 to Ensemble, which is obviously a slightly ridiculous thing to have tried to do, but as a naive user, it doesn't seem that weird. They will ban people's IPs, which maybe we don't want that to happen to our users. And it's unclear to me like how frequent these massive queries are compared to other things that are going wrong. But it's one thing they noted to me was that they are kind of keeping an eye on queries that come via biomaRt. They also say that a replacement to biomaRt with the big B, big M is coming at the moment. The last thing they said to me was spring 2020, but I've been hearing this for at least 3 years now. And so I will believe it when I really see it at the moment. Although they did also say that they will try and let me know about this variable renaming thing if they do it in the future before release time, because this one basically crept up and broke packages overnight. So it would be nice to have at least a little bit of warning that it's going to happen. And so then I have a few kind of suggestions for things that I might want to do in the midterm if there's not a replacement and these kinds of issues continue. It might be that my solutions have already solved it. There doesn't seem to be quite so many at the moment, but that may be just because it's summer, for instance. So I could impose limits on how big the queries could be, how many values you can put in a filter or something. I don't know exactly how you would draw that line, but maybe 17 million exceeds that. I could also actively try and promote other things we have in Bioconductor, right? So we have the Ensemble DB packages, we have the Organism DB packages. Ensemble themselves have a REST API, they have like bulk download via FTP and things. Sometimes those may be much more appropriate than using biomaRt. And I don't know whether I should stop queries happening or just update the vignette and say, perhaps you don't want to do this. I don't know whether people really read the vignette, but I'd be open to suggestions as to sort of maybe the best way of trying to direct people towards using other resources. Alternatively, I guess I could just use them under the hood, right? I could try and catch certain behavior from users and do it automatically and silently so that existing scripts and syntax works exactly the same. I think this will probably be quite difficult to implement in practice. But again, if that seems like a sensible way of not breaking existing code, then perhaps that's something that I should look at. And alternatively, maybe we want to actually develop specific tools for the most common things people do. It seems to me, based on, well, talking to people, questions I get, and then a poll I put on Twitter, the vast majority of people that use it just want to convert ID types. There's tons and tons of information in Ensemble, but all they want is a mapping between A and B. And we have many other resources for doing this. So again, maybe we either want to promote them somehow or present them to users and that kind of thing. But I'd be very happy to get any feedback on this kind of thing, any discussion about it. So thanks very much. You can talk now.
Hello, Mike. This is Thiago. Can you hear me? Can you hear me?
Yep.
Okay, great. So I'm one of the users that normally my package breaks because of biomaRt, at least in the last 2 months. And I think the work we have done the last months were amazing. So thanks very much for that. The first thing I have a question about the mirror thing. I had a problem, like I was using the main one, and then when it was down, I had to get another mirror. Now you told about the geolocation. What happened if like the Asia one is down? Do you redirect it?
So I guess if you get redirected to a mirror which is then down, it will just fail. I don't do any checking. If it fails kind of thing. So the redirection happens server side, and I follow the first redirection, and then whatever comes back from there is the answer, is the result kind of thing. So yeah, I know the Asia one has, it's been a bit more temperamental than some of the others at the moment. I don't have a solution to that. So the redirection happens without any help on my part. Which mirror you get redirected to is definitely done by Ensemble's team.
Okay. Great.
But yeah, I have done at the moment. So by turning it on, by default, if you put nothing in as a host, you will get redirected to your whatever they send you to. But if you explicitly say US West or www or whatever, that's the one you'll go to. So if the person writing the code explicitly says they want to go to a particular server, that's where they'll end up. If they don't, you end up wherever Ensemble sends you.
Okay, great. I also received I know the data table package team, and also it seems I received an email from Hadley this week. It's an automatic email. So I think they're doing an inverse building of package. Mainly, they get all the packages that depends on there, and they try to compile. So I received an automatic message saying we're updating our package, and yours is going to be broken in 1 month. So I'm not sure how they did that. I'm not sure if Bioconductor is implementing something like that. But I think at least for the biomaRt might be quite interesting, since there is a lot of packages using it. If there is any change of variables, because I also had the problem with the entry engine ID, and it took me a while to understand why my code was breaking. So it might be something good for the developers to take a look, at least the base package. I'm sorry?
No, I was just going to say that that's our Devel mailing list, right? And that's also why we have BioC Devel, right? So, when it all crashes down, it doesn't affect release, and we have a couple of months to fix it. I mean, I guess it would be nice to have specific emails, but I mean, I get those emails from the mailing list anyway, so it's not a big deal. That's just what I think.
Yeah,
So
yeah.
one problem is that one issue is that the variable renaming isn't something I did. It happens on the server side, right? So as soon as an ensemble release came out, they changed this thing. Even the release version of biomaRt and all of the other packages using it now just had the wrong string in, basically. So there was no concept of a Devel release or anything like that. It just broke overnight, essentially. This has happened before with other data sets that had weird characters in and things, and there's not very much you can do to prep for it.
Okay. Mike, with respect to the question about the caching, how I get around this in some of my workflows. So for example, the simple single cell workflow uses BioC file cache to pull down stuff, right? And how I make sure that it's always rebuilt is I set the cache to the working directory of the vignettes directory. So it caches it inside the package as it's getting built, rather than using the sort of system cache. And that means that when the package finishes building, it just gets deleted, and then it starts all over again during the next build.
Right. Okay. Cool. Yeah, because I wasn't sure if it was a good idea to even have the cache on the build system, right? And maybe you want it there because you kind of it means packages will more likely build rather than failing arbitrarily do things out of their control. But I guess
so what you can do is that when you test your own package, when you test biomaRt itself, you basically turn off the cache or make the cache local, so that biomaRt itself checks whether it is working correctly, internally. But when other packages build, they can use the system cache, and and if you leave that as the default, that's just what will happen. So the other packages will be like packages that depend on biomaRt will just happily work even when the server fails, and when the server fails, only biomaRt itself will fail. But that's okay because you just do it again, right? That's not a big deal.
Yeah. I'm fine with that. I'm expecting it to fail. Okay, this also answers my question about whether the cache would persist between builds and that kind of thing, because I don't really know much about how the build system works under the hood, sort of thing. Just what you get reported back.
Hello. So the cache should remain from build to build and daily builds. We don't clear the cache out, so as long as it wasn't using like a temporary location, it should persist.
Excellent. I can't remember what location it uses, but but it's not the temp file, so.
It should, and I can always check on that if you.
Cool. Excellent. And what do people think about the ideas of, I don't know, either promoting other resources, right? I mean, I guess for instance, Ensemble DB is used. That's downloaded quite a lot already. Other people are using it, but should I have a message that pops up that says maybe this isn't the tool for the job, or should I just ignore it and hope something better comes along in the future? I'm not really sure what my strategy should be here.
Mike, one expedient approach might be whenever you get an error from the server via biomaRt, you suggest some alternatives.
Yeah.
Right, and then given the rate of how many errors we get, right, you'll just naturally push people towards these other systems. But if they really want to use biomaRt, then they can they'll just have to sort of stick with the errors and keep on going.
Yeah, agreed. Like a try catch and then some kind of message of what alternative possible solutions could be. And like Aaron said, eventually they would filter out if they keep getting that message and have to use the other one. Eventually they learn or not.
Yep. Okay, yeah, that sounds eminently sensible.
And then just supplement it in the documentation, which people usually don't read anyway, but you never know. And then that should be okay, I think. I don't think there's a need to actively push people off because that freaks them out.
Yeah, and I guess I could hide things under the hood and use other tools to keep syntax the same, but that feels like an awful lot of work for a tool that might not last that long. Okay, if that's enough discussion on biomaRt, then Aaron, do you want to present your update?
How do I do this? How do I? Okay. Whoops. Yeah. Okay. Yep. Oops. Crap. Shit. Okay. Okay. How did you do this, Mike? You just.
So I have a button at the bottom of the screen.
Okay. I see. Well, I just want to open PowerPoint. And this technology is just... Okay, give me a second. I just got to figure out how to use my computer. Okay, all right. Okay, I have PowerPoint open, and now I got to share my screen. And okay, good. All right. Can people see this?
Yes.
All right, great. Yeah, okay. Yep. Okay. Yeah, so I'll talk a bit about the single cell updates for all the single cell packages that I maintain, which is a few of the sort of more basic packages in the BioC stack. Right. So one of the big things that we did over the past few months was to introduce this concept of alternative experiments. So the idea here is that when you do a single-cell RNA-seq experiment, sometimes you're not just doing RNA-seq. Sometimes you're doing stuff like site-seq, where you have antibody tags. So you have counts for surface markers. Sometimes you're doing CRISPR-seq experiments, where you have counts for CRISPR, the CRISPR guides. And I guess the most obvious example is that if you ever have an experiment with spike-ins, you also have counts for the spike-in transcripts. These aren't endogenous features. These are just sort of these aren't endogenous transcripts. They're just other features. And we wanted a way to represent these more formally in the single-cell experiment class compared to our current approach, which is just to stick everything in a big matrix and have them all mingle together in downstream analyses. So what we came up with was this idea of a alternative experiment. And to cut a long story short, all this is they are nested summarized experiment objects that are parallel to the columns of the original single cell experiment. So I've got this little snippet of code here, and what it basically does is it pulls in a single cell experiment object. So that's this big SCE object. I don't know if you can see my mouse, but I'm looking at it now. So that's this SCE that gets pulled down from experiment hub using this Lun spike and data command. And within this SCE object, there is an alternative experiment for the ERCC spike in transcripts. So what this is basically another summarized experiment object that's within the SCE object. And if you use this Alt-Exp SCE ERCC command, you can get out the summarized experiment that contains this ERCC, that contains the counts for the ERCCs. And you can do sort of other stuff like, get all the, so here I've just got one, or I think I have two alternative experiments, but you can have, any number of alternative experiments. So you can have stuff, if you do this mega experiment that has, counts for endogenous genes and spike in transcripts and, site-seq, so counts for antibody tags and counts for CRISPR guides, you can have all this information in a single object as separate alternative experiments. So this assumes that we have the same set of samples in all matrices, which is usually which is the assumption that we that we can rely on when we do these types of highly multiplexed experiments. And some of you might notice that this is effectively just a very lightweight alternative to a multi-assay experiment. The reason why we didn't go with a multi-assay experiment was because it was, well, for starters, it wasn't quite necessary for what we needed because we always knew the samples would match up, and we didn't want to have to switch all our analysis code to be compatible with a multi-assay experiment if that was effectively the new basic data structure for handling these types of experiments. So we wanted people to be able to store these alternative features as these alternative experiments in a single cell experiment object, and then if they didn't care about these alternative features, they could just keep on using the single cell experiment object as if it was, as if those alternative features weren't there. So by keeping with a single cell experiment, we ensure that it's back compatible and it's relatively easy to understand. And the entire motivation for this, so part of the motivation for this was to allow users to represent these objects or represent this type of experiment in this class. But from a developer perspective, part of it was actually to avoid confusion between feature types that you store in the same matrix. So one of the problems, at least for me, when I was dealing with spiking data was that whenever I wanted to do an analysis on a count matrix that was stored in a single cell experiment, I had to always remember to subset out the spiking transcripts so that my analysis would only work on the endogenous genes. And that was a real pain because sometimes I forgot. And then the code would just do silly things. So with this alternative experiment idea, the sort of main assay in the body of the single cell experiment object, that will only ever hold endogenous genes, and that makes it a lot easier to write functions for, because if you just need to work on the default is to just work on the endogenous genes, so now the matrix just holds endogenous genes, and now it's all nice and easy. And if you do need spike-ins, that's usually not the default behavior, so you just go and your function can just go and pick up the spike-ins from the alternative experiment slot. And that is making it easier to do the default thing is good. Right. So that's the alternative experiment. Another thing that we've been doing quite a bit is to do a lot of refactoring of the Scran and Skater packages, which have varying levels of dependencies throughout the rest of the Bioconductor ecosystem. So Skater, for example, is now responsive to this concept of an alternative experiment. So if you do something like plot t-SNE, and you give it a single cell experiment object, and you ask it to color by a feature that is in an alternative experiment, Skater is smart enough, or plot t-SNE is smart enough to know to search the alternative experiments for this alternative feature to do the coloring of points by. Similarly, if you do something like calculate the per cell QC metrics, this function is smart enough to know that it needs to go into the alternative experiments and compute metrics for those features as well. Scran similarly uses the spiking data from alternative experiments as well, and that makes life a lot easier because it means that if I have functions that don't need to use the spiking data, it just does the default thing of just extracting out the main count matrix from the single cell experiment. It no longer needs to subset out the spike-ins in order to do the rest of the analysis. And in functions that do need to use the spike-ins, it's just as easy as specifying which alternative experiment, in this case, the one named ERCC, holds the spike-in data. So that's a much more logical progression of how data should be retrieved from the object. For other developers who might be interested in using this alternative experiment feature, as a general rule, functions only need they only need special behavior for alternative experiments if they need to use the fact that an object is an alternative experiment. So what I mean by that is that if the alternative experiment itself is a summarized experiment object. So if you're just doing a calculation that just needs the data from a summarize from the data in an alternative experiment, you can just call whatever function you want to do the analysis with on the single cell experiment or the summarized experiment that is extracted with the alternative experiment. So there's no extracted with the alt-exp getter. So there's no, so in a lot of cases, there's no need to have special, like if you're writing a function that works on a single cell experiment, there's no need to have special behavior for the alternative experiment objects. There's only need to have special behavior in very specific cases where, for example, I need to know that the ERCCs are an alternative experiment of the SCE in order to use the ERCCs to model the technical variability of the endogenous genes. So it's when you have to combine information between the alternative experiment and the main experiment, that's where you need a function. That's where your function needs to have sort of special behavior for that for these alternative experiments. Another sort of thing that I've tried to follow is that is for both of these packages to follow the golden rule, where every method, insofar as possible, every method can be used on both the raw matrices and single cell experiments. So that means that if you're following the bioconductor ecosystem and you're following the workflows and you're using a single cell experiment, then everything's easy. But if you're outside of that and you want to, you're not using single cell experiments, but you still want to use the analytical methods, you can do that directly on raw matrices that you supply. So you don't have to do this hassle of wrapping everything in a single cell experiment just to use the functions. And that's quite important to make sure that these packages can be used not just by end users who might be using single cell experiments, but also by other developers who might be passing in raw matrices. Yeah. So a third thing that we've done is this is a singular package. So this is a package for automatic cell type annotation of single cell RNA-seq data. How to use it is pretty straightforward. So we have some reference data. In this case, we have some inbuilt reference data sets, but you can use whatever. You can use your own, any reference that you have labels for. So we get a reference data set. We pass it into this singular command with this reference, and you give it a test data set that contains your test data. And then that gives you out a set of predictions for each individual cells, saying what it thinks each individual cell is, right? So, in this case, the human primary cell atlas data contains labels of cell types. I can't remember them all, but neurons and T cells and whatever. And then if you pass that in, you can get basically cell type predictions for each of your cells in my_SCE. So yesterday we put this into the queue, and that's pretty vague, but it's somewhere in the queue there. So hopefully we can get it through, and we've done some refactoring for so that it's actually quite fast. So if you give it 100,000 cells, on one call we get it done in 16 minutes. And part of this is quite easily parallelizable. So I'd imagine if you throw more cores at it, it just goes faster. Of course, so this was a work by this was done by a bunch of people. So Devere was the original author of this method, and Dan, Jared, and Fred have been contributing to getting this bioconductor ready. So if anyone, so please play around with this and let me know what you feel. Okay, so the final bit is this book. Right. Okay. So most topics in this book are completed. So quality control. Yeah. So I've listed the stuff that we've done. This QC thing isn't quite done. It still needs some plots, but everything else is more or less done. In progress. I'm working on the data integration right now, but there is still quite a lot of work to do, and we need bodies to do this stuff. So if I can't remember who's an author on this paper, but if you're an author on this paper, please, pick one of these to-do topics and help out, because right now it's just me and Rob and Kevin, and it's, I basically spend, my evenings writing this book, and it's really boring. So, if everyone chips in with a bit, we can get it done in a week. And then that would make me a lot happier. Yeah, okay. And that's it from me. And let me turn on my video. Is someone talking and I just can't hear them, or what? Okay.
We were waiting Turn on your machine.
Right. Yeah. Okay. Yeah.
The book, is there a link to it? So the channel is like a private channel in Slack, which you have to be invited, and it's about the review of the paper, but not the book. But now it's like it's gone a little bit off topic. So I just wonder whether it's something that you want to share in a different channel or make it open.
Right. Yeah. I don't know if Rob is here. Rob, are you here? I guess he isn't then if he's not speaking up. Oh wait, Steph is here.
Yeah, I don't think Rob is here. At least I don't see him on the list of people. So that's a great question. So originally the channel was private and it still is. I'm not quite sure why it should be. I'm happy to make it public. But basically this orchestrating single cell analysis book has taken on a life of its own, and it's fantastic. And so, as Aaron said, we could definitely use all the bodies we can in terms of helping develop contributions towards it. The paper itself is under review, so that's kind of on its own right now. But if you want to contribute to the book, the online book, you can for sure. Does that help?
Yeah, it was probably private because of all the trash talking we were doing during the review process.
Yeah, maybe. You have to get your frustrations out somehow.
Yeah. Unfortunately, because we don't keep track of we don't have the paid plan on Slack. All that trash talk has disappeared. So
Right.
It should be fine to make it public now.
Yeah. One question I had about the book is, are we including? Are we only focusing on single cell sequencing, or should we include maybe single nucleus sequencing as well?
I mean, we could, right? I mean, it's basically the same thing, yeah. Cell, nucleus, they're both sort of round things. Just stuff it in.
Maybe you can
I'm
read
asking-
it
Sorry,
in its own chapter. Yeah, sorry. I was just
some
thinking.
chapters, yeah. Okay. I'm starting to
Same.
work with a lot of single nucleus data because people are working with cells that are very hard to dissociate, and so they're just basically capturing the nucleus right now. So I've just started looking at this data, but I'm trying to identify people who have sort of like compared this to what they're getting out of just regular single cell RNA-seq data, and then if there are nuances to the QC or things that we're going to have to address towards that. That's the only reason I ask.
Yeah, because as Aaron said, it's probably the same analysis overall, but then probably if everything is described for the regular cells, then having just an extra chapter saying special consideration for nuclei.
Yeah, that was one thing I was going to bring up.
Just do SED S stroke cell stroke nuclei.
Said what? Okay, right. Okay, said. Sorry.
Oh wait, that doesn't work for nucleus actually. Okay, all right, okay.
So are we doing nuclei type and nuclei states as well? Or but yeah, maybe one thing for the book that I wanted to bring up is considering how many chapters and how big it is already, it's kind of a bit hard as for newcomers, I guess, to have a feel for how to structure a chapter. What should it? I mean, the first thing I noticed is that every chapter starts with motivations, which is a good thing, saying, okay, why is this chapter here? But once you get past that, it's a bit hard to just know, all right, so what do I start with? Should I pick up an example from the from a workflow, or should I make up my own example? So I think Aaron mentioned maybe writing some golden rules for how to contribute a chapter. I think there is something in the README already to
yeah,
sort of...
there is something. Where's my pen? Okay. Yes, there is something. It's I mean, I haven't written it much because I haven't gotten any contributions. So I guess that's a bit of a catch-22, right? And I won't get contributions until I write it. I mean, for the time being, I think probably the most expedient way is just to have people coordinate with us. And I think if we can make the Oscar review channel public and rename it to something else, like just the Oscar book or something, that would help. I don't think there's that many. Well, I might be pleasantly surprised, but I don't think there's that many contributors that, it would be a bother to help to help them on a one-on-one basis. I mean, if there are that many contributors, then that's also great, and I'll put in the effort to make it more systematic. But until that time.
Yeah, I'm
happy
right
to
now.
figure out how to make the channel public and then rename it to Oscar Book. I think that's a good idea. So. I mean, the book has really just taken on a whole life of its own. It's like its own project now, and so it should very much be its own thing.
To ask a question about the software, this alt exp stuff.
Mhm.
Is there potential for generalizing that beyond single cell? It seems specific to single
Yeah.
cell.
Yeah. I mean, we could move it somewhere else. It just happens that single cell experiment was the thing I had write access on. So
I mean, if it proves useful in the single cell space, it might be worth considering generalization.
Is the rule that the samples are the same but the features are different?
Sample, yes, that's right. Yep. So if you really want to get into the nitty gritty, how it is actually implemented is through some dark S4 magic where I've stuffed it into the internal code data as a transposed summarized experiment object. No one ever has to know this, but well, now you do. So that's how it's implemented. So that's how it enforces the parallel, the sort of parallel nature with the samples. That each of the alternative experiments is parallel to the samples of the alternative experiments is parallel to the samples of the main experiment.
Great having Michael sit beside you there.
Yeah, he made this face too when I told him about it.
Yeah, I think that the whole point of this call is that so that we do understand how you've implemented that, right? Like that's one of the outcomes that I kind of want from these chats is for other people to have some idea of how the various structures are work and are built and that kind of thing, right? Like it's interesting, it's cool, whether it's black magic or not. I think it's really helpful for other people to at least have seen it done and know whether it's possible and whether it's sensible. Maybe others have thoughts on that too, but like that's actually one of the outcomes I want is to hear the kind of nitty gritty of how things have been implemented.
I agree.
Right, we are in the developers forum after all, right? The more technical, the better to some level.
So
I should have put in
Aaron,
my slide about interesting C++ bugs then.
So Aaron, Tim, Trish has a question on the chat. He says, when some single cells have measurements and others not, is this better or worse than MAE, and in which situations?
So right, so the alternative experiment concept is strictly when all cells have set there are no missing cells in any particular set of alternative features. So if you do have missing cells, and the sort of example that leaps to mind is something like what was it? Sorry, something like RNA-seq plus methylation or something like that. In that case, it seems more pertinent to have that as an MAE because that data structure can handle the fact that you have basically non-identical sample sets between the objects. And in that specific case of transcriptome and methylation, usually you wouldn't be doing, at least from what I understand it, usually you wouldn't be doing analyses on both of them at the same time until basically the very end where you want to integrate the results. So at least like for the routine stuff like quality control, looking at t-SNE plots, clustering, using both levels of that information would be a dedicated step that, something like Moffat does or something like that. And sort of, and then in that case, it's fine because it's sort of worth the hassle of, working on a single cell experiment, working on something that holds the transcriptome data and working on something that holds the methylation data separately and just waiting until the end to integrate them together. Here it's more of a, for the SEE alternative experiment use cases, it's more of a case of, there's a lot of points where they have to play together at the same time. But, the workflow in which you use where there's, for example, the endogenous genes and spike and transcripts have to play together is also more or less the same workflow that you use when you just have endogenous genes. And we wanted to make it simple for people to just easily switch between the two modes and not have to worry about, as soon as you have spike-ins or as soon as you have site-seq transcripts, you have to switch to an MAE. So that's mostly the motivation there. and why the sort of alternative experiment, why we had that instead of having an MAE.
Yeah, it seems like a coercion makes sense, right? The what I was actually thinking of when I was talking about this was for VDJ measurements, or TCR PCR measurements on the same cells, and sometimes the results don't make very much sense. Sometimes one of the arms is there and the other isn't. But I guess it makes sense, your point, that the QC and the switching back and forth between assays, probably the QC can almost always be done independently, or you set a hard cutoff where if it's not detected as a cell, then just throw it out of all of the assays. That makes sense. And then a late coercion would make sense too.
I have a question about the singular package, Aaron.
Yep.
So what format does the reference data need to be in? What do you need to have in the reference data?
So the reference data can be any, basically count matrix or even normalized expression matrix, PPMs.
And you have to have every cell, you have to assume every cell type is being included in that reference data?
Okay. So, right. So, yes. Right. So that depends on what the aim of your annotation will be. So if I have, say, for example, if I know my input data is T cells, but I want to classify them down to, particular subsets of T cells, then it's fine for the reference data to just include T cells with just very high resolution labels, right? So that's okay. If your aim is to just have, if you've got a bunch of something you don't know and you want to classify it, then yeah, you're probably going to need something. You're probably going to need a reference that has, all cell types that one could feasibly expect. So some of the inbuilt references are fairly broad. So the human cell atlas one, the human primary cell atlas is pretty, it's got quite a few labels, or quite a few general labels, so that might work in general. But yeah, I mean, you can build your own reference and that's fine, because we allow you to do that. There's just this train singular command that'll just train everything.
And these labels, they can be computational labels? I mean, right? I'm assuming they're computational labels.
What do you mean by that?
So what labels are you talking about when you talk about the reference data?
Right. So these are effectively any arbitrary labels that you're interested in.
Okay.
Right. So they can be as simple as this is a TB macrophage mast cell, or they can be cluster 1, 2, 3, 4. As long as they're meaningful to you, right? They can be any labels that you want. For example, if you're trying to match up two data sets, then having them as clusters, just numbered clusters, might be sufficient. Yeah. Because you don't as long as you can figure out what the clusters are in the end, you just want to match them up, identify which cell corresponds to which cluster in another data set. But if you're doing something like, I want to know what cell type it is, then the labels better be cell types, right?
Right. Thanks.
Cool. So we have 5 minutes left, and I, at least for the first meeting, want to try and stick to my stated hour. So one thing I do want to discuss is how we pick topics going forward, right? So this time was basically me panicking that I hadn't done anything with 2 days to go. Aaron stuck his head up above the parapet and asked the question, so he got roped in. And then obviously I did the other presentation myself. So I have a list of sort of topics that were generated by me, Martin, and Michael, which I guess we thought were interesting, certainly a few months ago when we wrote the list. But I would be curious to know whether people want me to just try and assign them and find a speaker based upon my own sort of expectations or whatever, put the whole list out and let people vote and choose. Do people have an opinion about this? Did it work with, I think it worked with 2 topics for one session, which basically filled the hour. What do people think?
I like the idea of 2 topics in an hour.
Yeah, it gives time for a short presentation and then the question and answer.
Yeah. And what about picking topics? Like, obviously I can just dictate, essentially. But if I can't find somebody to present, you're going to get a lot of me giving presentations on topics.
I think there should be like maybe a suggestion box of topics. Maybe we could discuss in the last 5 minutes of every meeting like this which topics we should present next time.
There should also probably be a sign-up ability for somebody to sign up if they have something that they want to discuss.
Yeah, so I mean, hopefully going forward, I'm not going to do it 36 hours before the meeting. I'm going to put a little bit more forethought into the agenda and that kind of thing. Yeah, so I guess probably the easiest way if people actually just have a topic they want to present is to message it to me, and then we'll sort of put it out, I guess, in the Slack channel or something as to whether it's generally applicable or whether it's literally just you that's interested in it. I suspect that's not going to be the case. Yeah, obviously I'm totally happy for volunteers to present.
Seems like the Slack channel would be a good place to coordinate, get a smaller selection of possible topics for the next one, let people sign up for it or see what the interest is.
Yeah, so as I say, I had a list which I guess was in the sort of PDF I put in the very first email around about this, but I'll take that list and put it in the Slack channel explicitly. So at least to give a starting point for potential topics, that kind of thing. And the plan is to basically do this every month, so approximately the middle of September. Maybe not the 15th, that's my birthday, but we'll see. Does anybody have a pressing topic about now? Like, as in, suggest now?
I'm currently working on submitting a data package to Experiment Hub, and there's some discussion on Big Data Rep about naming conventions for these packages, and I'd be curious to having a larger conversation about if we should impose some structure conventions of naming packages.
Yeah, that was quite an epic conversation, wasn't it? You disappeared for an hour and there must have been like 100 messages when you got back.
Yes, I had a meeting and 1 hour later there were 100 messages.
Yeah.
So really sparked conversation.
Yeah, so I also
went away for 5 minutes for lunch and I think there was 20. Like, yeah, it was like
Yeah, I also felt that there was a potential for basically boiling down probably mostly Casper's frustrations with file formats and delayed array and all sorts of different things into some sort of objectives for how we can kind of
well, I think file formats is a different thing. So just naming conventions of packages on its own is like a topic, but then also whether or not we want to impose some kind of structure restriction of what the object is when it gets submitted to Experiment Hub.
Okay, I will add it to the list of potential topics. Let's not start the discussion now with 1 minute, just to right. Whatever o'clock where you are. But thank you for the suggestion. In which case, thanks very much, Aaron, for presenting, and thank you everybody else for turning up. Hopefully it's been useful and informative. I'd love to get people's feedback, and hopefully see you in a month.
Thanks, Mike.
We're putting it together, Mike.
Thanks, Mike.
Thank you. Goodbye.
Thank you, Mike.
Bye.
Bye.
Thank you, Mike.